Please join me in welcoming Kania from Flagship Ventures.
All right, so this is the first of these events, and therefore my primary role is to make all of you, hopefully, but at least most of you, excited about coming back for the second event that they try to hold. In pursuit of that, I'm going to try to do a couple of things in a rather undisciplined way, for which I apologize. I'm going to try to tell you a little bit about my world as a venture capitalist with Flagship Ventures. Try to illustrate that in a way that makes it maybe more relevant by telling you 6 stories. I shouldn't tell you 6 stories, but I've got 6 stories that I want to tell you because I think they're really relevant to the Mayo. Some of the Mayo's directly involved in. Some I think are evocative of what you guys can do here and why I'm excited, and I think you should be excited about this experiment, this effort here. And then I want to introduce Roland Harris, CEO of Medita Metrics. Again, because I think he's a special guy, but also, and Roland, forgive me for this, because I think he's evocative of, hey, anybody can be an entrepreneur. And when you hear his background,
and not necessarily do it well.
See, he laughs. So that's what I want to do. I'm going to fly through some things because, again, 6 stories is too much to tell you, but examples are sometimes much more powerful than words. And I guess I also want to, before I start, just also reflect and acknowledge something that Jeff said, which is that the challenge of starting also applies to venture capitalists and venture capital firms. And I stand here today in a firm that I think has achieved a lot and is pretty significant in its field, but really would not exist here without the support and deep partnership with the Mayo, Harry Hoffman and his group over the years. So you need to know that that makes me heavily indebted to you all through them. And to the extent that I can do anything in support of any of you here, you as Mayo have more than earned that right to ask that of me. So thank you for your support in getting us started. And I'm in fact talking about the challenge of that, and whether it's a venture firm or a company, it's not easy. Okay? So listen, we're talking about translation of innovation, and therefore sometimes you say, what is the most significant thing that's happened in terms of innovation over the last 100 years, over the past 50 years? You get a group like this together, start asking people questions. You usually get a list like this, running from the internet to biotechnology to personal computers, mobile phones. That's the sort of typical list. I would submit to you that actually the biggest innovation is the startup venture, which far more than most big company attempts, institutional attempts to take ideas and translate them into products and things that change the world, the startup venture has been totally transformative. And as examples of that, I would point out all the things on the left are actually delivered to us by one or more startup companies of the sort that are listed on the right. The Googles, the Apples, the Genentechs, and it's not just entrepreneurship in the for-profit world. The entrepreneurship in the not-for-profit world is just truly spectacular and inspiring through things like Teach for America and Partisan Health. So that's kind of what we're all here for, supporting, trying to do more of. But in fact, it's not easy. And we at Flagship have the privilege of participating in a small way in that ecosystem. As Jeff mentioned, as a firm, we're about a 12, 13-year-old firm. We're based in Boston, Cambridge actually, in the very interesting ecosystem of companies and academic institutions and hospitals and CROs that exist there. At this point, we've got 4 venture funds that we manage, totaling about $900 million, and we really specialize and focus on entrepreneurial innovation and trying to translate that into things that create value for the world and for us and our investors. So here's a bunch of headshots of our team. We've got a core group of 7 of us. Can't really tell much from that, but I've actually founded Flagship, co-founded it with my partner, Nubar Fain, a long-time extraordinary entrepreneur whose board I had the pleasure of serving on for about a dozen years before we founded Flagship together. The firm, we actually think about it in terms of two component parts. The first is Flagship Ventures, which operates as a classic early stage, hands-on partnering with entrepreneurs, venture capital firm that invests anywhere from $500,000 to seed something up to $20 million to support the growth of a venture. And then we've got something called Venture Labs that I'll describe briefly and in a bit more detail. That is our attempt to try to be a little bit more systematic, a little bit more thoughtful, a little bit more institutional in starting companies, in starting great companies. And we're doing some unique things there. It's not directly analogous to what you've got here, but maybe there's some ideas that we can take away from that would be helpful here. So again, we're focused in healthcare and in clean technology, all based on life sciences technology. Within that, we focus on the areas of therapeutic devices, therapeutic platforms, medical devices, and various services and technologies that can improve the outcomes and reduce costs. And you can ignore the words here, but the punchline is that the bar for starting companies is very high in today's world, or any world, as it should be. And we really are trying to come up with ideas and approaches that are compellingly better than what exists today, because if they aren't compellingly better, you know, there's so many challenges in starting a company that you might as well not even embark on it. So they got to be differentiated and compellingly better. However, how do you tell something that's compellingly better? It turns out to be really hard, and I was having this discussion with some of the folks here earlier, because honestly, any of the things that we have started getting excited about, at the point in time we got excited about, at least half the people we talked to thought they were totally absurd. So Einstein is absolutely right. If at first the idea is not absurd, then there's no hope for it. Unfortunately, absurd ideas can also be absurd. So against that backdrop, we operate off of what is really a fairly simple formula for what we do on the venture capital side. We try to work with field-leading academics and clinicians, catching the ball from them, if you will, using some of the products of their ingenuity and instincts. We combine them with dynamic entrepreneurial leaders in the context of a team that creates a bit of a culture that's special and is often a mission to change the world. Again, there's got to be an underlying breakthrough approach in technology there. It's got to go after a major, important, unsolved problem. And then you sprinkle onto that small amounts or usually large amounts of money. And that's kind of the formula. So let me take that formula and play it through a couple examples in rapid fire here. Don't expect you to get much from these other than some high-level visions of company starting. Agios is a company that we founded with 3 academics in 2007, Lou Cantley, Tack Mack, and Craig Thompson. And they were 3 academics who had been thinking about the area of metabolism for a period of time. They met together having a drink at a conference and realized that independently they'd been thinking about the same thing from different angles and got very excited about it and came to us and told us about it. Sort of what they realized was that the observation that Otto Warburg had made in 1923 that cancer cells metabolize more rapidly had actually reached a point where it was actionable from a drug development point of view. That there was this potential thing called the metabolome that was different than the transcriptome or the genome, and you could actually start mining the metabolome for asking, are there targets that we can attack that change the tendency of cancer cells to hypermetabolize and therefore treat cancer? That in essence was what they thought they understood. So we went from November putting $2 million in to doing a lot of work, and there's another backstory about all the work that was done between November and June. But in June, we got confident enough that we were willing to, a group of 3 venture firms coming together, put up $35 million tranched into the company. Published about 9 months later, well, First, then reached out and got David Schenckheim, who's an extraordinary MD, researcher, clinician, drug developer who had been at Millennium and Genentech, brought him in as CEO from the West Coast. Shortly thereafter, published a Nature paper changing the way people thought about the metabolism in cancer cells and taking something that people thought was actually a good thing and describing how it was a bad thing. With that in hand, we went out and got a $130 million upfront cash commitment from Celgene to support the company, which was great, because all of a sudden we had money for this far-out idea. Went out and raised $78 million on top of that, published recently an important Nature paper, sort of further defining what this field could be, and now have 85 employees, expect to be in the clinic late this year or early next year, based on 2 lead programs that have fundamentally the opportunity to target something that people had thought was actually a tumor suppressor target gene, but actually turns out to be an oncogene. So pivotal insight that could change fairly broad-based treatments for a bunch of cancers. So that's an Agios story. 3 academics with an insight and a process in an entrepreneurial system. Something similar, but fast forward, because it's in process, you can watch this with us. In October last year, we put up $2 million again, backing 3 extraordinary guys who kind of self-assembled in and around Metro Boston out of the Whitehead and Dana-Farber. Rick Young, Jay Grabner, and Nathaniel Gray. They had the observation that a whole new set of tools that they knew how to use would enable you to try to understand transcription regulation in a very different way. They were so excited about this, they were literally giggling when they came in to describe this to us. Couldn't figure out what the business would be, but they were really excited. And we, working with them, became convinced that they had come up with an approach that would allow them to identify specific transcriptional targets and simultaneously develop a small molecule drug and a biomarker to go with them so you could run efficient clinical trials and do things that no one had done before. This morning, in Cell, they published the first papers describing this, so you can go online and read them. And it's kind of, we think, a big deal. They've identified something called super enhancers, which in the gene regulation field are a massive simplifying assumption. Gene regulation is a complicated field with all these things that nobody understood, but they've identified super enhancers which sort of allow you to take complexity and reduce it to simplicity. Over the last 4 months, prior to publication of the paper, we went out and identified and patented, dozens of these super enhancers. There's a limited number of them. And yesterday closed on a $28 million financing tranche to support the growth of a company to pursue what I think, when you read about it in Cell and some of the press that's coming out about it, is a transformative discovery in the field of transcription regulation, which is a key to controlling cancers and other diseases. So kind of a big deal. Who knows what the company will become, but this is sort of academics to guys like us, to companies that we hope will change the world. I'm going to take you to two very different stories. How are we doing on time, Jeff? Okay. To show you, because those are in process and it sounds glamorous. Now I'm going to tell you 2 beginning to year-end stories and give you a sense of what that looks like. In 1987, we gave $200,000 to an entrepreneur that I would encourage you to get out here because he's a deep friend of the Mayo, Stan Lapidus, to start a company called Cytek. And we teamed up with a couple of academics out of Tufts to start thinking about the field of cervical cancer. And the observation was that in the 1940s, cervical cancer was the deadliest cancer in women, but there was this thing called the Pap smear that had had a huge impact on it. And had really dropped the cancer rate down, but Pap smears weren't perfect. And about 40% of positive slides were missed, and this was sort of documented out there. So could we do anything about it? And Stan Lapidus was a machine vision expert, and we tried to say, could we bring computerized imaging to reading Pap smears, okay? So we started a company, we went down that pathway, we built a beautiful automated instrument, microscope of the sort that I'm sure you've seen here. It produced beautiful images of cells on a slide to help the technicians zero in on specific parts of slides that might be suspect, to help them do a better job. We ran a clinical trial on it, and whoops, it was rather uninspiring. So we were sort of at a dead end. Well, we sort of looked at the reason why, and the reason why is that the smeared slide turns out to be almost impossible to read because it's layers of mucus and blood in 3 dimensions. So we said, gee, no one's been able to do anything about that. What can we do about that? Stan came up with a breakthrough that allowed him to build a device that actually made slides out of the samples collected in pap smears called the FinPrep processor. You should really hear the story. It's a great story sometime. It made beautiful slides. You can't see them there, but all the cells, a representative sample of cells go in round spot sizes. When you look at them under a microscope, it's a beautiful thing to look at, easy to read. And we got strong patents in sample prep. We did some tests. The cytotechnicians did a better job screening it. We had a lot of tussles with the FDA, but ultimately they approved it as the first major improvement in pap smear testing in 50 years. We did a public offering in 1996. And then went on after an initially very challenging commercialization phase to achieve 80% market share in the US in pap smear testing. So big deal. It's the most profitable company in the history of the diagnostic industry. It was acquired for $6 billion in 2007, and you can see the prototypes of the first FinPrep machine down at the Smithsonian. So a nice story, but for those of you who think, gee, sounds easy, there were about 5 times during that endeavor where I was fairly convinced the company is going to die tomorrow. Bring it to Exact Sciences, which has a deep connection with Mayo because the academic co-founder, the original, the dominant one here is David Ahlquist, your own David Ahlquist, an extraordinary guy. Again, Stan Lapidus. It was what do we do after cervical cancer? And this time started thinking about colorectal cancer. And the problem is, how do you find a lesion in the colon, given that it's 6 foot long and looped around? And in the 1990s, we started in 1995. The way people did it, and actually the dominant methodology still, was the colonoscopy, which is a brilliant solution, except that it's kind of gruesome when you think about screening an entire asymptomatic population with a relatively rare disease with a 6 foot pipe up their backside. So the question we started exploring with David Ahlquist was there anything better we could do? And David had a bunch of ideas, we had a bunch of ideas. The starting idea was using something called Sammy's stool over here on the left. And Sammy collects cells from the entire length of the colon. We hypothesized that the cells might be preserved. If we could only preserve them and analyze them, we might be able to do a pap smear for the stool for the colon. So that's what we set out to do. Whoops, it didn't work. Bad news because we had a bit of momentum at that point. So we sat around with David and said, what do we do? Well, David had already started working on some stuff that Bert Vogelstein for Johns Hopkins had reported in 1992, which was something called a Vogelgram, which is the way DNA mutations occur in cells in the colon, leading to colorectal cancer. Beautiful, almost Nobel Prize winning discovery of the origins of colorectal cancer. So we said, hey, let's go get Bert and his team at Johns Hopkins to join us as scientific co-founders. Then we'll go out and detect DNA in stool as a way of detecting colorectal cancer. We went and got a whole bunch of discoveries and patents in the DNA in stool and how do you detect DNA. We then were like rocking and rolling, raised a couple of rounds, did a lot of things. Public in 2001. In 2002, and I'll take you all the way up here to today, so you got to be patient, I'm sorry. Got the largest partnership that anyone had ever gotten in the diagnostics industry with a company called LabCorp, big lab company known to most of us. Then in 2003, unblinded the major clinical trial that we had done, and I remember being with David Ahlquist at a conference when we heard what the results were, and they were really disappointing. They were They were worse than we expected. They weren't what we thought the test could perform because we had screwed up some things in how we ran this major $15 million trial. But nonetheless, they were what they were. And we demonstrated strong superiority of DNA testing to the fecal occult blood test, but not to colonoscopy. So it was a disappointment. Nonetheless, because we didn't have any other choice, we tried to start selling that because it was better than fecal occult blood. Tried to get inclusion in guidelines, went and developed a second generation test, but by 2007, we were pretty much out of money and out of luck with all these paths that we've gone down. And in the winter of 2008, David Ahlquist and a couple of folks from Mayo came to visit to me and said, you know, gee, we really believe in this. Is there a way we can keep this company going or maybe do something to restart Exact Sciences because this is a good idea? And that kind of rattled around in my mind, particularly as we then finally, 6 years after we started, got American Cancer Society approval. But this is a public company with 4 employees at this point because it's about out of money with a stock under $1. And at that point, you know, I got mad because it didn't make any sense to me. This should be great, but it was not. And we figured out that this IP that we filed for everything other than colorectal cancer had value, and managed to persuade Gentime to give us $25 million for us. Took that $25 million, went and conducted a national search, got a guy named Kevin Conroy in Madison, Wisconsin to step in as being the CEO. Kevin got the opportunity, decided it was a challenge he wanted to take on, and started on a 3rd generation test, and he understood the value of Mayo, and the first trip he paid was to come out to Mayo and reignite the partnership with Mayo. So with Ahlquist here, they came up with a sort of a next generation screening approach using a bunch of different markers. Pretty simple yes/no test result that would refer you to colonoscopy if it's positive. Have a decent form factor for it. Ran some clinical studies on it that have produced really exciting results. And this on the top, you probably can't see, is with 1,000 patients, 98% detection of cancer, and as astonishing, 57% detection of precancer. So really could change medicine, assuming that this holds in a large, rigorous, randomized, controlled clinical trial, which is underway right now. It's an $18 million trial, 12,500 patients. It finished enrolling in November, and in the next week, the top-line results will be reported. This is 17 or 18 years after this company was founded. An envelope's going to be opened, and we're going to find out what the results are. Here's exact stock price since it's low in April of 2008. I won't show you where it was. It sort of started up over here, but I don't like to look at that. It's gone from 37 cents to 10.70 today. It'll go way back down if the trial doesn't work. It'll go up if the trial does work. It's open the envelope. It's an important development in colorectal cancer. I believe in it. I hope it works. I hope it works for Ahlquist and the Mayo as well. So with those sorts of experiences, it won't surprise you that when we started Flagship, we also started thinking about the entrepreneurial process, the entrepreneurial innovation process. Was there anything we could do to make it a little less chaotic, a little less erratic? And we created something called Venture Labs, which we call our Flagship Innovation Foundry, to try to create companies based on breakthrough ideas that might have slightly increased probability of winning by being disciplined in spending a little money very aggressively early on to prove things out, and then working with these companies as they grow. It's turned into kind of a real entity, part of Flagship now. Its mission today, it's a group of people, it's actually quite impressive, is actually to create transformative companies that change industries. And it's a group of people within Flagship that focus on this, plus a network of advisors, including some folks out here. And then some processes that we think are proprietary, but like most processes, they're just common sense. And there's some things that you might be able to think about for some of the work you do here. But basically, it's sort of an evolutionary process for testing company hypotheses that mirrors what we actually see happening in the companies we work with. So here's a team of people that work there. I show that, and you can't see them there, but there are a lot of young people here. I want you to keep that in mind. This is a game of the young in addition to the old and experienced. So young people are good. Since we started this in 2000, we started 25 companies. A bunch haven't worked. A bunch have been shut down before they became companies. But some marvelous things have emerged out of this. The way we do it is to be pretty liberal in something we call explorations, and I'll tell you exactly what it is in a minute. But it's sort of the exploration. We had an idea presented to us by a researcher at Children's Hospital. I have a way to alter the genome of cells using messenger RNA, and I think it can produce stem cells that doesn't involve embryonic stem cells. I'd like to start a company there. What do you think? What do you think about it? So we start explorations around either inventions or unsolved needs. Some of the explorations result in things we call proto companies, which get seed capital, $200,000 to test out ideas. And after a process of testing the ideas in proto company form that I inelegantly call throwing a company up against the sharpest rock you can find. So we try to find the best critics in the world, have the company make its pitch to those critics, and if they survive the wrestling match, they can go on and live another day. And that's the tough role to startup companies. Then they can become a startup company. And we do that pretty well, I think, and you can't see this, but how can you make renewable fuels without needing sugar? Can you use CO2? We started a company that's got, raised all sorts of money at fancy valuations called Joule, that has solar collectors that collect sunlight and use CO2 and blue-green algae to produce fuel. It's a pretty neat, potentially transformative idea. Something called 7 Sense, trying to think about, are there different ways to collect blood that might transform the billion-dollar vacuum cleaner business, because that's been around forever and just feels like it may not be the most efficient way, particularly given the small sample sizes you need these days. So explorations ultimately leading to new ventures, but the process is actually one that is not linear at all. And here's a company that has some contact with Mayo, in part because Mayo is among the world's best people in something called the microbiome. You won't be able to see the print here, but in 2007, people started understanding that the genetic diversity in our guts surpassed the genetic diversity in our cells, and that the genetic makeup of a fatter person's gut was different than the genetic makeup of a thinner person's gut, and that if you took and switched them in mice, fatter became thinner, thinner became fatter. It's fairly astonishing, very unexpected. We started thinking about it then, and we sort of launched an exploration in mid-2007 into the microbiome. That's 5+ years ago, thinking about what you might be able to do with it. We first identified a diagnostic opportunity we thought was a big deal. We pursued it, put $200,000 in, did some experiments, did some market research, we abandoned it. Didn't work out. We then identified a therapeutic based on some work we did in mice that we thought would be important for something called Crohn's disease. Got all excited about that, did an exploration, put some money, created a proto company, then found that there was no correlation between mouse and people based on some work we did in the background. We shut that down. Okay, so we're 3 years into the microbiome. We've tried a couple ideas out. We've got nothing yet. We run something with some MBAs and PhDs during our summer called the Flagship Fellows Program. And one of them brought to our attention this thing called fecal transplantation, which has been publicized a lot right now, but it's known that if you take anyone's feces and put it into a patient with severe C. diff, untreatable by antibiotics, you can virtually cure 100% of them, and it was not believed because it was all anecdotal until somebody did the randomized controlled study published in the New England Journal. So everybody's gotten excited about this. The reason that occurs is, of course, because of the microbiome. You're repopulating somebody's microbiome. So in mid-2010, we started thinking about how you build a company around that. So feces to curing C. diff. We started wondering if we could do something that was not quite like putting feces into another person and create a biological equivalent of the microbiome. We hired a CSO, we built a company around that, we filed some patents. And so today we've got this thing called Ceres, which we've invested in. We've brought in another set of folks that is trying to, in fact, develop a synthetic FDA approvable version of a feces transplant to treat acute C. diff, and we think we can roll on other similar diseases, assuming we're successful in that. So again, Ceres today is trying to define what we call the ecobiotics. It's a way of treating diseases caused by a dysbiosis in the flora and fauna in your gut. Medically, scientifically, it should be a big deal. It's a new field, and we're proud to be there with a company, early stage company there. We're hoping to partner with some of your clinicians here who are doing breakthrough work in that area. Final story, then I'm going to turn it over to Roland. A company called Moderna started in December 2010, and I mentioned this before when a young scientist at Children's Hospital named Derek Rossi found a way to get messenger RNA into cells, modify their genome to effectively turn them into stem cells. It was a really intriguing idea because it was another end around embryonic stem cells. Weren't sure what we'd do with it, but we put a little bit of money in and started an exploration in it. We rapidly realized that if you could actually use messenger RNA, not produce an immune response, and get a cell to change its genome, you could actually get a cell to express a compound. Which actually turns out to be interesting because if you can get a human cell to express a compound, you may have a way of getting a human cell to express any drug you want, because you can actually send in the codes to produce almost any drug. So we got quite excited about that, did some lab experiments which actually proved it was plausible, got a great CEO to step in about a year after we founded it, did some more experiments, went out and raised 2 rounds of capital, and then last month announced a mega deal with AstraZeneca, whereby AstraZeneca put up $240 million cash in the barrelhead. So we have a company right now that almost has as much money as a venture fund does, sitting in its bank account to go after this audacious idea that you can take messenger RNA, inject it into the bloodstream more locally, have it reprogram cells to produce drugs. We've got patents on this way of producing drugs that go around every patent for every drug that's that comprise the pharmaceutical industry. There are lots of challenges with the idea. We could all come up with them. But if it in fact works, and with $240 million, we have time and money to push hard at it. And by the way, for that, AstraZeneca got rights to cardiovascular, all cardiovascular, rights to some cancers, no equity, and they have agreed to pay us $180 million in milestones as we go through a couple of non-human primate studies and stuff we're doing right now. So, I guess what I would like to have you think about all that is that the game of starting companies is a rough and tumble world. It's not easy. I could tell you a bunch of failure stories as well, but they aren't as much fun for me to tell. So you got to have a very high bar for what you're willing to invest your time, your energy, your reputation in. I would encourage you, and I see this at many places, rather than thinking an idea comes out full-baked and that it reflects what you want, think about the Moderna example. Use the idea not as the basis for building a company, but as a basis for thinking about a field. Seeing if you can reconceive how people think about an area and what's done currently. This is the power of our exploration, and it's a very low cost, very powerful thing. Explicitly work together, bring people from multiple institutions. Mayo is a fabulous place. You've got some of the best researchers, clinicians in the world here, but it's not the only place. And we live in Boston where we've got access to all these wonderful people. We almost never do a startup just with people in our little regional area, because it's a big world, and bringing people together, you gather IP, you gather a brand, you gather so many things that you don't get when you just keep yourself insular. It's very hard for any academic center to do. Be patient with the process and time frame. Sometimes you have to move fast, but usually you have a lot more time than you think, because if it's a race, you're probably going to lose. You're just raising money is hard, and all the inefficiencies of a startup. So be patient with both the process and the time frames involved, because starting these companies up is often like pushing on a rope. You're trying to move it, but you're pushing against a rope. Youthful talent and vigor. You need experience, but it's amazing what you get if you send out a smart young person with a credit card to go talk to a bunch of people in the field. They come back with the most amazing insights by connecting people and dots that would have not otherwise connected. And so you've got a lot of that here. You use that in your processes of individual companies and in broadly in entrepreneurship as a Mayo. Stan Lapidus is a big promoter of focus. The way you actually achieve things is to focus. You got to start out with a bigger lens, but as you move forward, you got to become a domain expert, and the people that we back end up knowing more about their field than anybody else in the world. And because the best ideas can get screwed up by the wrong leadership, you got to have a high bar about entrepreneurial leadership, which is a great intro to Roland Harris. But I'll leave you with one parting thought, that probably the most important thing, and Roland will relate to this. And if you can't see it, you're looking at some sort of carnivorous bird swallowing a frog. But it's hard to know who's going to win. I bet on the frog. So don't ever give up.